Hello, I am Marco Ruggiero, although, as has been the case for some time now, the voice is not my original one with its Italian accent, but a synthetic one generated by artificial intelligence with the goal of improving the clarity of exposition. Today I want to tell you a curious fact, a story that emerged from our research on the different types of microbial strains present in Bravo. In particular, a story concerning a plasmid. Plasmids are small pieces of circular DNA that bacteria use to exchange information aimed at preserving their well-being, and consequently ours since we host them. We discovered that in the microscopic world of Bravo, where time is not marked by hours but by the rhythms of fermentation, an epoch takes place daily that unites the peaks of the Swiss Alps with the vast steppes of Mongolia. This is not just the chronicle of a biological process, but the narrative of an unprecedented genetic migration, revealed by the potential of the axiom microbiome array. The advanced research tool we used, capable of mapping the complexity of over 12,000 microbial species. At the center of this adventure is our Swiss product fermented with milk and colostrum, Bravo, an ecosystem of extraordinary biodiversity that hosts a literal fleet of 48 natural plasmids. These small rings of DNA act as true treasure chests of vital information for bacteria and for us. The adventure begins with the identification of an unexpected traveler, the plasmid PLCA36. This fragment of genetic material originally belongs to Lactobacillus casei strain Zhang, a microorganism isolated in kumis, the traditional beverage of Mongolian nomadic riders obtained from mare's milk. The scientific surprise lies in a paradox precisely detected by the data. While the plasmid PLCA36 is present in significant quantities in the Swiss product, with an observed percentage exceeding 55%, the chromosomal DNA of the Mongolian bacterium that should host it is totally absent. It is as if we arrived at a scene where the luggage is intact, but the traveler has vanished into thin air. However, this exotic transmigration is not a coincidence, but the result of a refined bacterial survival strategy known as horizontal gene transfer. In a competitive environment like that of kefir grains, bacteria do not just reproduce, they actively exchange their plasmids to adapt to changing environmental conditions. The plasmid PLCA36 brings with it advanced technological equipment. It possesses the RELBE toxin antitoxin system, which acts as a growth modulator allowing the cell to survive in conditions of severe nutritional stress, and a series of conjugation genes called TRIAA that facilitate its passage to new hosts. This survival kit has moved through countless generations, finally finding a new home in European ferments and contributing to the resilience of the entire ecosystem. The utility of this molecular adventure extends far beyond bacterial biology, as these genetic elements are closely linked to the promotion of human health and longevity, defined as healthy life expectancy, or HALE. Other traveling companions in this product, such as the plasmids of Lactobacillus helveticus or Lactococcus lactis, encode for essential functions ranging from the production of bioactive peptides against hypertension to the creation of molecular electron sinks necessary for bioenergetics and detoxification from heavy metals. The story of Bravo's bacteria teaches us that natural evolution operates with a sophistication that predates human genetic engineering by millennia, orchestrating a synergy between different species to preserve and transmit the most advantageous genetic information for life. I like to think that when I take my Bravo, usually at dinner, like an excellent dessert yogurt, inside there is genetic information that, from the horses of the Mongolian steppes, made its way along the routes of Asia and Europe until reaching the Swiss Alps, and from there to Arizona, to end up in my body and help me maintain the health of my microbiome and, consequently, my entire being. It seems a bit like the migration path of my human genetic ancestors, but we'll talk about that another time.